I'd like to welcome everyone to today's webinar with Dr. Michael Steele, who is an assistant professor at the University of Alberta. To give a short bibliography or bibliographic introduction to Dr. Steele, he completed his bachelor's and master's of science at the University of Guelph, and then after completion of his master's went into work in industry. For a year, he worked over in China serving as a dairy feed consultant for a year over there, and then he returned back to Canada and served as the head dairy nutritionist for Master Feed in Canada itself. And he did that for 2 years and decided, you know, maybe we'll come back to academia, and he completed a PhD in animal and poultry sciences also at the University of Guelph. And the focus of his thesis or dissertation was the rumen epithelial changes that occur during grain-induced ruminal acidosis in cattle. After completing his PhD, Dr. Steele went on to conduct a postdoctoral fellowship and then also worked as a research scientist for 2 years before taking on his current position at the University of Alberta as an assistant professor. Dr. Steele had a very successful research career here. In the last 5 years alone, he's authored or co-authored 28 peer-reviewed journal articles and also published 31 conference papers and abstracts in the past 5 years alone. And with that, gosh, I don't know where else to begin, but turn it over to Dr. Steele.
Yeah, thank you very much for this opportunity. I really appreciate the opportunity to speak to the entire ADSA group, but specifically to graduate students. So the format of my presentation, I'd like to give probably a little bit more detailed background of who I am and how I evolved to be here presenting today, as well as getting into the topic of nutritional regulation of gastrointestinal development in calves, followed by some of my current hypotheses and research themes that I'd like to share with everyone. So as mentioned before, my background, I come from a dairy farm in Ontario, Canada. I have 30 years of experience milking cows, and here's some evidence of that in the bottom left of this screen. I thought I was going to become a geneticist when I was young because I was very interested in breeding cows. This is one of the cows that I bred with my father that became 4-time world champion at Madison, but I ended up going to the University of Guelph and took fundamentals of nutrition in my second year, and it completely changed my life. I really enjoyed nutritional biochemistry and ended up doing a master's degree through coursework with Dr. John Kent, where I got to learn nutrition as well as molecular biology. I then had the opportunity to consult in China, and that was one of the most interesting years of my life, and I highly value that. But I had to make the decision whether to live in China long-term or to move back to North America. So I moved back and I became the head dairy nutritionist for a company in Canada called Master Feeds, which is now owned by Alltech. And my thirst for knowledge really brought me back to doing a PhD. And this was a pretty easy decision because I knew that I always wanted to do a PhD. I was lucky enough to get accepted into Dr. Brian McBride's lab at the University of Guelph and was fully funded by the Dairy Farmers of Ontario through a PhD scholarship. It's one of the best moves of my life, and it really changed. I knew at this point that I wanted to be a dairy researcher. So my PhD, as mentioned by Ben, was looking at the structural and functional changes of the rumen epithelium during grain-induced ruminal acidosis. And here's some TEMs of rumen tissue before and after grain challenges. But the bulk of my work was really characterizing the structural adaptations of the rumen epithelium. So I used a variety of microscopic techniques. And I also did a lot of transcriptomic profiling. And I identified different metabolic and structural genes that were being regulated through the responses of acidosis. And I also identified growth factor mediators during acidosis as well as during the transition period. So it was a lot of fun, and I must say that my years in grad school were the best years of my life when it comes to being productive and learning as much as I can. I really valued that experience. I had the opportunity to work 2 years after my PhD for Nutreco Canada Agresearch, now known as TROW, Nutrition Agresearch. And that was a great opportunity working for a multinational company such as Nutreco. So on a daily basis, I was working with several different brands within this company, and I learned a lot about the practical applications of research. It was an amazing experience for me. And I was fortunate enough to work on several different research projects within industry related to ruminant gut health, and also calf gut health and early life nutritional programming. So at this stage, I became very interested in the calf about 2 to 3 years ago, and this has really dictated a lot of my research program. But I was very fortunate during this time working in private industry. I was also supervising students, and that's what really motivated me. I came to the realization that I think my talents and what really motivates me in life is having students maximize their complete potential, and it made me think that I should move to academics. So a year and a half ago, I joined the University of Alberta, and I'm in the Department of Agricultural, Food, and Nutritional Science. We have quite a few dairy professors within our group, including Dr. Masahito Oba, Barim Amataj, Dr. Leilu Guan, Dr. Zikwan Wang, and Devakar Ambrose. So we all have our own skill sets, and we work pretty closely together on many projects. And last year alone, we had over 50 peer-reviewed publications related to dairy. So I've been very fortunate to be put into this very productive environment at the University of Alberta. I've had a lot of success with all of the dairy researchers thus far. Our research farm is right in the middle of Edmonton, Alberta. So we're completely surrounded by 1 million people. We have a 146 cow tie stall with a surgical suite. And my first year at the University of Alberta, we put in new calf technologies. This is just one example of a new calf technology we put in, and it's called the calf rail, where you can actually individually feed calves, feed calves using automation in individual pens, as you can see from this video. So this has been a lot of fun. We actually have a conference June 8th this year where we'll have Dr. Bob James from Virginia Tech, Dr. Jim Drackley from the University of Illinois, talking about automated feeding and calf feeding. And we also have some group-housed technologies that are brand new as well, where we have the ability to look at water consumption, all feed consumption, dry feed, hay consumption. And we've also involved some more enrichments of the environment, such as this brush here. So that's how I evolved to get in this position. That's what I've been doing at the University of Alberta. So I've been here for 1.5 years. But I'd like to get to the bulk of this presentation, where I'd like to discuss the dietary factors influencing the development of the ruminant gastrointestinal tract focused on the dairy calf. So I'd like to discuss some calf management trends that I've seen through my work in the industry as well as in academics, and discuss some recent research I've been doing related to pre-weaning, looking at neonatal development as well as feeding plane, and then looking at some recent papers that I published related to weaning strategy and also post-weaning. So I'd like to start my formal presentation by this graph that I was fortunate enough to grab from Alex Bach. And just looking at the overall feed costs are obviously the highest during this pre-weaning period. It's very difficult sometimes to really show the value of even doubling the overall price of feed and the cost of feeding in this pre-weaning period. But I think there's a lot of opportunity to maximize overall production, also production efficiency if you do invest during this period. I'm also very interested in this post-weaning period, and I'll get to that later on in the presentation. But what I'd like to say is that the feed efficiency is actually incredibly efficient post-weaning, and I think that there's opportunities to take advantage of that feed efficiency at that time. But overall, it's very expensive to feed calves and heifers through this period, but most of the cost is coming in the first 4 months of life. In fact, some cases you can reach 25 to 50% of the total cost in the first 4 months of life. The total investment of rearing a replacement heifer being $2,500. I'm a large fan of a lot of the work coming from Cornell University and other universities looking at early life dietary regimes and their influence on lifetime productivity. I had the opportunity in industry to work with Fernando Sobran, also communicate with Mike Van Amberg. And I think that this research looking at lifetime productivity related to pre-weaning average daily gain is very interesting. A lot of people might debate about it, but I've yet to see a response that's negative with respect to accelerating growth or feeding more milk during the pre-weaning period and lifetime production. There might be some studies that don't show significance. There's many studies showing numerical differences, but a lot of studies are showing the significance there. But again, I've yet to find one study showing a negative response on lifetime milk production, which makes me very interested in the potential of this type of research. Another thing that I've noticed through my work in industry, and this is just example of one farm that I know of during their weekly treatments of antibiotics. And I noticed that in industry, we are overusing antibiotics during this pre-weaning period. And that really initiated a lot of interest for me in this area. So this will be a large topic for me in the future, in particular, many experiments in 2017 and 2018. And I'm interested in the impact of using antibiotics in early life on gut physiology and gut function later on in life. And I think it's very interesting to note that antibiotic use pre-weaning has been associated with decreased lifetime milk production. So I think that this overuse could have long-term impacts. And I think the mortality by age as well as farm size, and this is just data from NOMS, still tell us, even the updated surveys that I've been reading in Canada as well as the US, is that we still have a quite significant mortality and morbidity issue within the pre-weaned period and also during weaning. And some of the numbers are reaching up to 10% for mortality. And a lot of this morbidity is coming from gut health challenges. And that's really the focus of mine for the next 5 years. Although I'll be doing some transition cow work, probably 80% of my research program will be looking at how the gut develops and what nutritional and management factors can we manipulate to improve over gut health. So I'd like to start by discussing gastrointestinal development. I think it's important to note that it actually starts before birth, and there's some elegant work out of the Cornell University in the 50s, just showing all the compartments are formed by this 3rd month. We really don't know that much when it comes to prenatal development of the gut of the calf, but I think that there's a lot of research groups that will be focused on this area for the next 5 years. So hopefully they'll have more insight in that area. To date, we think from a microbiological standpoint that the gastrointestinal tract is devoid of live microorganisms at birth. There is some debate in medical research with respect to that, but at this point, that's what we think. And immediately after birth, there's a rapid inoculation of the gastrointestinal tract from the dam as well as the environment. Firstly, let's say in the rumen, it starts to be aerobic, but within 12 to 24 hours, it becomes an anaerobic environment. There's been some publications showing that even what's happening during the pre-ruminant phase, or the microbials that are colonizing the rumen, still stay there and are very similar to what you would find in a mature ruminant, which is very interesting to me. I've been very interested in received some funding in my first year looking at colostrum in overall development. I think it's important to note that colostrum is a large collection of probiotics and prebiotics such as oligosaccharides. And I think our focus of research over the last 2 to 3 decades has been purely on passive transfer of immunoglobulins. But my interest is more in some of the other bioactives, or how you can manipulate some of the some colostrum nutritionally to get a better response. This is a publication that was in the Journal of Dairy Science, or an experiment that was just published in 2015 out of the University of Alberta. And it was done by Dr. Guan's lab. And it just compared heat treating colostrum compared to fresh colostrum or calves receiving no colostrum. No colostrum will be the NC here. This is fresh colostrum, this is heat treated colostrum. And just that treatment alone can impact the overall proportion of bifidobacterium within the gut at 6 hours as well as 12 hours of life. And I'm very interested in taking this research really far and going deeper into what are these mechanisms at play that are influencing these fractions within colostrum that can ultimately impact what bacteria are colonizing the gut in early life of a calf. I've also noticed through my work in industry that colostrum feeding is typically really focused on the passive transfer, and often calves are abruptly transitioned to a whole milk or a milk replacer even after the first feeding. I think that in nature that doesn't happen like this, and there's a tremendous amount of transition within this first week that I think we need to account for and could potentially improve overall gut health of a calf. I've really been a fan of the work out of Germany by Dr. Harold Hammond's lab, looking at the different proportions of different bioactives within not only colostrum, which is this first milking, but also the second, third, fourth, and fifth milking, and comparing that to mature milk. Not only does the fat and protein, it's still elevated, but a lot of these bioactives, such as insulin and insulin-like growth factor, are still evident within this transition milk. I'm very interested in looking at duration of colostrum feeding and the duration of feeding some of these growth factors and the impact on overall gut development, microbiology, and health. And I'll be doing an experiment looking at duration starting in September of this year. I think a lot of the work recently, especially out of Germany, has been focused on what IGF-1 and insulin are doing and where they're being taken up by the gut in early life when it's present in colostrum. And what they've noticed, even with this increasing the duration of feeding, is that you get more mucosal growth. You'll also have an improved absorptive capacity of the small intestine. Their focus was really looking at glucose transporters, and they found very little difference there. As you can see through these, they were using laser scanning confocal microscopy here to analyze the expression of escaloop. So I think that there's a lot of opportunity here to dive deeper, but my interests are more in gut structure, transcriptomics, as well as the microbiology of it. With respect to after feeding colostrum, I think there's still a lot that we can learn, not just from the pre-weaning period, but in particular in the first week of life. I was really a big fan of this paper by Papillae that was published in 2014, just looking at the incredible variation that you can see in calves that are offered ad-lib intake to milk on day 4 of life. Typically in industry, we're feeding between 4 and 6 liters of milk in early life, but as you can see, if calves are allowed to consume whatever they wish, they can consume a lot more. I think that this is pretty interesting with respect to looking at accelerated feeding plans, but also in the first weeks of life, and not just waiting until the 3rd or 4th week of life to get up to 8 to 10 liters of milk. But the problem with this is that a lot of people are hesitant of feeding these accelerated feeding programs when they're only feeding 2 times a day, just because the calf typically in this first period can nurse up to 12 times per day in the first weeks of life. And larger meals are fed less frequently. They can increase the risk of milk overflow into the rumen. And as you can see, it will, we can fill up this rumen, but we can fill up this abomasum, but some can overflow into this rumen. And we call that, it could cause ruminal acidosis and also decreased rate of passage and digestion. And these larger meal size may also be associated with reduced insulin sensitivity based on some recent papers from Alex Bach's group in Spain. So I became interested in abomasal emptying, and one of the first experiments that I conducted here at the University of Alberta was related to abomasal emptying. So here's just an x-ray of abomasum that's full of milk, as you can see. So I was wondering if we fed an accelerated program only 2 times a day, so let's say 4 liters per meal 2 times a day versus 2 liters 2 times a day, what would happen to abomasal emptying and how would that influence glucose metabolism? So I had the treatment of 8 liters or 4 liters per day in 2 meals per day. So again, the meals were 4 liters versus 2 liters 2 times a day. And I looked at abomasal emptying rates using acetaminophen as the marker. I noticed that these calves, even in early life, as early as 3 weeks of life, can reduce their emptying rate if they're fed larger meals. And that went on, we looked at glucose tolerance using a glucose tolerance test. We noticed no differences between glucose areas under the curve as well as insulin area under the curve in this particular experiment, where these animals were offered this accelerated feeding program 2 times a day from the first week of life. So this was one of my first graduate students, Jaden McPherson, who started a 1.5 years ago, and she did this experiment actually in the Netherlands with my collaborators in Nutreco, which is in the southern part of the Netherlands. Very beautiful place if you have the opportunity to visit. And I think a lot of these technologies, where I'm going right now with this type of research is looking at what's happening in early life, in particular the first 2 weeks of life. And I'm doing some experiments in our facility right now, looking at different meal sizes and also feeding frequency. And I'm using even automated technology to look at these. So I have an experiment running right now where I'm looking at calves in the first weeks of life being fed on a calf rail. One of my undergraduate and master students are looking at that. Over the last 2 years, I've focused a lot of my efforts in weaning, and I was very interested in, with accelerated feeding programs, some of the challenges that are involved with respect to weaning, since calves will not be consuming as much starter. And really, weaning decreases, a problem during weaning can definitely elevate morbidity and mortality. And with these accelerated feeding programs, we need to pay very close attention to how we're weaning these animals with a step-down protocol that's proper. And I think this transition is very accelerated, and I'm very interested in this dramatic change in overall rumen volume and the overall function of the forestomach during this weaning period. It's a wonderful biological model to study gut development and adaptations. And I've just always been fascinated by this very abrupt weaning that we have in dairy when you compare it to beef. I come from a dairy farm. I will admit that I do also have some beef cows, and I've just been amazed by the complete difference biologically during this weaning period. We typically wean extremely early in dairy, and we wean abruptly, even if we have a 10-day step down. That would be classified as very abrupt compared to what happens in nature. I started one of my first experiments I did on a collaborating farm in Ontario in this field when I was working for Nutreco Canada Agresearch. And I just compared weaning age when calves are fed in accelerated feeding programs. So I've compared 6 versus 8 weeks at weaning. And the results on body weight were kind of interesting. With respect to the 6 weeks, there was a lag in overall growth post-weaning. But the ones that were weaned at 8 weeks didn't have a dramatic lag, and they ended up being 9% heavier and did not reduce their overall metabolizable energy intake during this period. As you can see from this graph, and this is a paper in the Journal of Dairy Science last year, show that these calves at 6 weeks that are being weaned, when they're being fed this accelerated program, just aren't able to adapt quickly and consume as much metabolizable energy compared to animals during this 8-week period. And this coincides with other results showing that there is something or some kind of clock in the animal where genes in the rumen can turn on and off around this 8-week mark or as early as 6 weeks. But I think that there's something going on with respect outside of nutrition based on this experiment and some recent results that will be published this year in Frontiers of Microbiology, where we've looked at the microbiology response in this particular experiment. I think it's interesting that in both treatments, weaning did increase ruminal short-chain fatty acids and blood BHBA and fecal starch, but these differences were very dramatic in the calves weaned at 6 weeks compared to 8 weeks. There was a bigger difference between these. This led me to another experiment that I conducted at Nutreco, and it was a step-down weaning protocol. And I wanted to just characterize a very severe step-down. So I did an abrupt step-down versus a gradual step-down. And here are the body weight responses with respect to the step-down treatment, which occurred at day 36. So these calves were being fed 9 liters 2 times a day, then they were stepped down to 4.5 liters at this stage. And there was a lag in the overall growth here. Compared to animals that were abruptly weaned at this period, there was definitely a larger lag here, but when it was all said and done at this day 54, there was no difference in overall body weight. And that carried on for months after. What I found interesting was the dramatic intakes, and I really wasn't familiar with intakes that could potentially go from around 250 grams to 2 kilograms within days, and before I started to study calves. And I really translated this into metabolizable energy for intake, and I was amazed that obviously during this weaning period of an abrupt weaning, it's going to be a dramatic reduction in overall metabolizable energy intake. But it is also quite severe even during a step-down protocol. So I think that there's a lot of opportunity here to improve our weaning strategies, even if it is a step-down. Some people would classify this as a step-down. It's a quite abrupt step-down, but it's commonly used on a lot of farms, at least in our area of the world. So I think that there's a lot more work that needs to be done here. In this particular experiment, I took all the animals at day 54 and did dissections. And I noticed very little differences in gross anatomy. There were some differences in overall forestomach weight when it was full. And the rumen full weight was definitely significant, but the empty weight, there was just a trend. And as for rumen morphology, I did different surface area measurements using micro-CT. I also did rumen papillae length and width measurements using a stereoscope, and I found no differences between these treatments. I'm sure if I took samples right at the day of weaning, I would speculate that there would be differences, but I think this goes to show how rapid this rumen is developing, especially when you have spikes in intake going from around 500 grams to over 2 kilograms in the matter of days. What I found really interesting about this particular experiment were the high levels of butyrate during weaning. We were reaching, in both treatments, levels around 25 millimolar during this post-weaning period, which is very high compared to other results we found in the literature. I also looked at the lower gut, and this is where I started to think maybe we should be looking more into this lower gut and not just focusing weaning studies on rumen development. So when I started looking at rumen development, my experience beforehand during my PhD was looking at mature cows during acidosis. So I was amazed. This is the first calf I ever dissected an image of it, where I saw a lot of these striations, what I would like to call hills and valleys here. You don't really see this in a mature ruminant. You see this in a developing ruminant from my experience. And if you take a cross-section of one of these, you'll notice that it's kind of like polyp-like structures, and from this polyp-like structure, you're seeing papillae that are being formed. What I was amazed by was a tremendous amount of variation within the length and also the width of these papillae. I was getting coefficients of variation of up to 40% within a region within an animal. So it made me think perhaps looking at length and width of a rumen papillae is not the best measurement for rumen development. So it led me to the notion of if what we're really interested in is looking at the actual surface area by looking at dimensions of rumen papillae, why don't we do 3D scans? So these were some of my first 3D scans of rumen tissue using micro-CT technology. What was interesting is that I found really no correlation between length or width and surface area. Typically, like on the right where you have small papillae, the surface area is actually just the same as something that, such as on the left where you have very long papillae. So I think looking at total surface area is pretty important. And I'm using this technique as I develop my research program and look more and more into the lower gut. I've been doing some recent scans on the duodenum, jejunum, and ileum so that we can look at very large segments and look at villi dimensions instead of papillae being in the rumen. But I'm interested in this technology and using it in a lot of my research program. I also became interested in barrier function at weaning, and I think that this hasn't been characterized very well. What really started this was seeing these elevated starch levels in fecal matter during weaning, especially post-weaning. And started feeding in calves has been associated with decreased expression of tight junctions throughout the lower gut as well. And that's some work out of the University of Alberta, Lei Luguan's lab. This is a study that I did with Dr. Greg Penner at the University of Saskatchewan. This is when I was with Nutreco. And we used chromium EDTA, or urinary chromium EDTA, as a marker of gut permeability. So we looked at calves at 14 days of age and 28 days of age before we imposed any treatments. We noticed a pretty big reduction in the gut permeability of the calf between 14 and 28 days. What was interesting is the weaned calves actually opened up their guts. At least there was a lot more chromium EDTA in these weaned calves at this period at day 42 compared to not weaned calves. And these were just a couple days after weaning when we did these measurements. So this shows me that there's a lot of differences with respect to gut permeability, and that weaning can really induce an increase in leaky gut in that ruminant. So a lot of my work right now is focused on characterizing the structural changes of these calves, and you'll be seeing that at ADSA this year. Some of my students will be presenting that data, as well as the gene expression changes within this study. Another area I'm very interested in is post-weaning. And I think it doesn't receive that much of attention, but the efficiency of these animals during this time post-weaning is incredible. Sometimes it's up to a 3.5 kilograms of feed to 1 kilogram of gain, which is the most efficient ruminant that I know of, and a lot more efficient than some actual feedlots in beef. This is a study I did with Dr. Trevor DeVries, and it was published last year in the Journal of Dairy Science, looking at feeding different dry TMRs, so very heavy concentrate diets post-weaning. So this is the third month post-weaning. We put these calves post-weaning on an 85% concentrate diet. The other 15% was chopped straw. We then transitioned half the calves to a 70% concentrate diet here, and then kept the other calves on an 85% concentrate diet, so the same diet, and then switched them to an all-silage diet, really just for fun, and we wanted to see how the dry matter intake would be reduced. What was really interesting is that during this age, we typically just feed concentrate between 2 and 3 kilograms, but as you can see from this graph, these calves can consume a lot more than what we typically give them if they're not on an ad lib feeding system of concentrate. This is the dry matter intake as a percentage of body weight. I was typically taught that this was around 2.5% when I was in grad school, but as you can see from this graph, it's actually quite high. It can reach up to 3.5%. I think this really goes to show that we might be limiting the overall intake during post-weaning. And as well, even if we're feeding an ad lib TMR or a high group TMR that we feed to our post-wean calves, we probably have NDF limiting overall intake. And the average daily gains of these calves are quite incredible. We were reaching some at 1.75 kilograms. And when we reduced it to 70%, it did reduce overall intake, but also gain, which was even reduced further post-weaning when we would put them on a silage diet. So I think NDF is really limiting intake post-weaning. And I think that you can make an argument that when it comes to growth, pre-weaning nutrition is very important, but also post-weaning nutrition is just as important. There's probably a lot more opportunity to gain more during this stage, so a lot of attention should be focused on this area. And I have an experiment starting in May looking at the interaction between pre- and post-weaning plane of nutrition. So I think this post-wean calf receives very little attention so far, but I think that we should really be focusing our efforts not just on the pre-wean calf, but also the post-wean calf that is often neglected. So my overall take-home messages for this presentation is that the neonatal phase is an area left undiscovered in calf nutrition, in my opinion. Elevated planes of nutrition can be fed early in life with a 2-time-a-day feeding scheme, as shown by some of my recent work with some of my grad students at University of Alberta. Weaning age and abruptness impact performance on high planes of nutrition. As well, weaning is also associated with reduced barrier function. And post-weaning nutrition is another area left undiscovered in calf nutrition. I'd like to conclude this presentation by talking about my research program over the next 5 years. So I've received 5 years of funding, and my focus will continue on this plane of nutrition, not only pre-weaning but also post-weaning, and looking at the interactions of that. Also, I've received funding to do colostrum research, and my interest there is the timing of colostrum feeding, the duration of colostrum feeding, and the bioactives within colostrum outside of immunoglobulins and how they're affecting the biology of the calf, in particular the microbiology and as well as the structure and function of the gut. I've also initiated some work in my interest in antimicrobials and what they're doing to the calf. So I have some experiments planned in 2017 and 2018 looking at waste milk feeding in addition to antibiotic feeding orally, as well as treatments and how that impacts gut function, microbiology, short and as well as long term. And I'm also looking at antimicrobial alternatives as well currently. Where I'd like to take my research program is looking at the maternal influence. So I have planned experiments looking at inoculations from mature animals into the pre-ruminant calf and looking at how that can affect microbiology of the gut as well as the metabolites within the gut, and thus signaling for growth. So that's really my research program, where I'm at right now and where I'm going in the next 5 years. The overall objective of this research program is to develop a better understanding of how early life nutrition and management factors commonly found and used in dairy production can impact gut development during this pre-weaning phase and also later on in life. And the overarching hypothesis for the next 5 years is that during the neonatal period or in early life, gut growth and barrier function signaling pathways in the ruminant are regulated by specific interactions among diet and microbiota that can impact biological outcomes later on in life. here's my team. I have 2 undergraduate students, Jade and Sarah, that will be presenting at ADSA this year. I have a PhD student, Young, who's co-supervised by Dr. Guan, looking at colostrum feeding schemes. I also have Amanda, who's doing an MSc, who's looking at colostrum feeding schemes. My farm technician, Jen, who does a lot of the work with the automation at the research station at the University of Alberta. My first graduate student, Jaden, who will be presenting again at ADSA. And I just added another student, Mariah, for a master's, and 2 postdocs have started with me this year. But I am still looking for more graduate students in positions. And what I'm really looking for are students that like to integrate classical nutrition. So I feel that I'm a classical nutritionist at heart, but I have a lot of experience in cell, molecular, and microbiology that I like integrating with this classical nutrition. So if you have any interest in that or know of any other students that would be interested in it, moving to Canada and moving to Alberta, I am looking for not only postdoctoral fellows, PhD students, and MSc students. And I'm looking for some that will start now, but I'm also looking for some that might be interested even years from now. I'd rather start the communication now with some of these students that might even want to join my lab even 4 years from now. I think it's really important to communicate with supervisors and get to know them before you would ever start a degree with them. So I'm very open to any communication. And feel free to email me if you have any interest or know of anyone that might be interested, and visit my website if you want to learn more about my lab. With that, I'd like to just have one final plug. If you're interested in knowing more about the University of Alberta and what we're doing, we are having a CAF conference this year, June 8th. It's between 10 and 3 p.m. And it will be on our campus right in Edmonton at the University of Alberta. And we're going to have Dr. Jim Drackley from the University of Illinois and Dr. Bob James from Virginia Tech speaking at this CAF conference. So if you have any interest in going to grad school, this would be a great opportunity for you to visit the University of Alberta to see if it's a good fit for you. With that, I'd like to thank all my colleagues and students at the University of Alberta and all my funding agencies, in particular NSERC, ALMA, Nutreco, Alberta Milk, and Lulliment, and all my students and postdocs. And I'd like to conclude with any questions. I'd be very happy to field them. Thank you.
Dr. Steele, I actually have a question for you myself. You started out working with mature cows in both a feed yard setting as well as in the lactating dairy animals, and you've migrated to calves. You've emphasized from your presentation that you think there's a lot of opportunity here. Do you see yourself going back to work with mature calves, or excuse me, mature cows in these production type settings? And if you think there's any significant opportunity there, could you elaborate on?
Yes, that's a great question. I think a lot of people are kind of surprised by this movement who are familiar with my PhD, but I really, during my PhD, noticed that I like very dramatic responses of the gut, and I was getting that with ruminal acidosis, but I think the most dramatic one is through weaning and in early life, so that really attracted me scientifically. On a very personal level, I had children at the same time that I switched to calves, and I noticed I was reading a lot of medical work in the field of early life nutrition, and that really inspired me to move in that direction because I saw a lot of opportunities in that field of doing very fundamental work that was completely applicable to what you would see in our field in dairy nutrition. So scientifically and personally, my interests were there, and I will always keep some form of lactating cow as well as transition cow research active, but for the next 5 years, I think it's very important to have focus, especially when you're starting, and I think that's where I see the greatest niche, at least within my research environment in Canada, but also my interest too. So I see myself, doing both in the future, but I'm very interested in this model of the calf right now, and that will be my focus. I also noticed a question from Mark Hill saying that I had audio problems in the late part of your presentation. Does early post-weaning nutrition have an impact on milk production later on in life? There's been very few studies showing that it does. have any impact. I don't think it's been very well researched, but I think that there's a lot more questions, especially with respect to some of the controversy in mammary gland development and overfeeding during the post-weaning period. So I'd like to dive deeper into that field. I won't have the biological replications in my future experiments to look at what post-weaning nutrition does to overall production and lifetime production, but I think that that's an area that I'd like to dive really deep into and not only look at the gut changes, but also what's happening in other tissues such as the mammary gland in greater detail. I'm just reading more of your questions, Dr. Hill. Through research topics, from your experiments in industry, were you able to pursue research topics that you were interested in, or was your research dictated by the company? I would say that's a really good question, and I'd like to compare and contrast research within the university as well as the industry environment. So they're both excellent. I was very fortunate with my position at Nutreco, which is now known as TROW. They were extremely supportive of me not only doing research for the company, for the purpose of the company, which is basically developing new products as well as feeding regimens to improve their overall profit, but also the profit of their partners and their overall customers. And they also allowed me to pursue more mechanistic research. So I was very fortunate in that regard to work for that company that supported me in that way. But I find with, and what really amazes me about industry research is that if you find something, there is a great potential that literally millions of cows could potentially be fed using your products or your feeding regimens in a very short period of time if that product does work and has some form of benefit for the producer. So to me, if that excites you, I would definitely consider a research career in industry. What I liked about academics, and it's just purely on a personal level, is that I like the interaction with students. They really motivate me. And you can have interaction in industry. Some of my colleagues do still supervise students, but it's more frequent, and there's a lot more students in the academic environment. And there is some freedom, a lot more freedom to not just pursue the goals of the business of the company, but you can also focus your research program on looking at more of your personal interests and where you think the industry requires more research. But again, a lot of my research here at the University of Alberta is funded by research, so by industry. So I think that when it's all said and done, there's not that large of a difference, and you can be extremely productive in both industry as well as academics. I think it's just your personal choice in if you like to interact more with students. What are the interactions among housing, hutches versus pens, and changes during weaning? I haven't looked at that field. I've just been focused on the nutritional side, but I would say that Alex Bach has done work in that area, but I think it would still be very preliminary. This is another question from Dr. Mark Hill. When in a calf's life does gut permeability pose problems? What increases the gut permeability? So I think that we need to define if gut permeability truly does pose a problem. If you look at other medical research, it does. There's some circumstances, such as in early life, where you want a gut that's a little bit more permeable so you could get more passive transfer. But at this point of time, we can just extrapolate from medical research saying that this could pose a problem, or even swine research where it's showing that there are a lot of problems with respect to a leaky gut during this weaning period. So we can't say for sure right now. What increases gut permeability? I think there's a lot of mechanisms at play, and that study that I just showed really was the beginning of it, just showing that there is a response. Now we have to dig deeper of what's increasing this gut permeability, and I would suspect that it's particular microbial as well as metabolite interactions in the lumen with this intestinal epithelium. And based on some of the microscopic images that I've been processing recently, I think a lot of it's happening not only in the small but also the large intestine, mainly in the duodenum based on the microscopic. as well as the proximal colon. Those are the particular periods where we're seeing changes in pre as well as in pre-weaned calves versus weaned calves. So as for what's increasing it, I would think that it would be microbial as well as metabolite responses that I would like to look deeper into, thus signaling into the intestinal epithelium to increase inflammatory actions and maybe reduce tight junctions. And another aspect, I think, with respect to permeability that's neglected a lot in particular in ruminants is looking at mucus thickness and the different mucus levels. To date, no one has characterized that in a calf or a cow, and that's a huge part of gut permeability and barrier function in a calf. Sonia, you mentioned the role of colostrum in gut development. Do you have recommendations or thoughts about how long calves should receive colostrum? Yeah, I actually do. In this particular time, I would, I'm really basing a lot of this work off a lot of my opinions on work from Germany, saying that extending this colostrum feeding or even feeding transition milk can improve the mucosal absorption capacity of calves. So as for now, I would recommend feeding colostrum or transition milk for as long as you can in a practical setting. There are limitations. You don't want to feed colostrum for the entire week, obviously. That would be extremely expensive, and I'm not sure if that would be good for a calf. But I would say feeding transition milk has been shown to improve gut health, and I'm interested in what particular components within this transition milk outside of what we focus on with colostrum, such as immunoglobulins, are contributing to that. So at this stage, I don't have a very clear recommendation, but I would encourage farmers to feed transition milk if they have that capacity on their farm, or feed more than just one meal of colostrum. In the experiment that I'll be conducting in September of this year, I'll be comparing one meal of colostrum, multiple meals of colostrum in the first 2 days, versus multiple meals plus a very small proportion of colostrum within a milk replacer being fed for an extended period of time. So hopefully I can have more answers at 2017 ADSA. Taylor, Dr. Steele, very interesting seminar. Thank you, Taylor. I just had a quick question regarding your opinion on feeding fresh colostrum. What are the downsides? I think this really is farm dependent, and I think that there's a lot of, if you have very clean colostrum collection protocols and you feel very confident and you've tested on multiple times that you're not having overgrowth of particular microbes, it could actually work just as well as feeding pasteurized colostrum. But if you have a lot of people working on your farm and there's a lot of opportunity for particular microbes to contaminate colostrum, I think it's a lot safer to pasteurize it. Granted, you're going to be altering some of the bioactives, which we truly haven't characterized in great detail. And the extent of that alteration, I think it's a balancing point. There could be benefits to not heat treating colostrum, but there could be benefits to heat treating colostrum based on the overall amount of bacteria, healthy and as well as bad bacteria within that colostrum, and how clean you are. So if you're concerned about the cleanliness of your colostrum, I would say to pasteurize. If not, I think there could be some bioactives within colostrum that could be being destroyed at this time when you pasteurize. But really, at this point, we do not know that, and we need to dive into it deeper. Dr. Gerald Huntington, do you see any role of the development of the lymphatic system in the leaky gut phenomena? No. At this point, there probably is, but to be honest, I haven't looked into this area in great detail. When I'm looking at leaky gut phenomena, I'm mainly looking at tight junctions as well as in the first day of some of the pinocytosis mechanisms is what I'm really interested in. But as for the lymphatic system, I haven't thought or have done any work in that area. So I apologize, I can't answer that in greater detail. Dan Shields, what about early rumen development and how would it impact life performance? I'm not sure what's meant by early rumen development, but I'm assuming in the first 4 weeks of life. I'm not really sure if it would make a large impact because I've noticed through the work where I'm actually delaying rumen development by feeding an accelerated milk feeding program. It doesn't take them that long to really adapt and have very little difference between animals that have had a larger step down or opportunity to eat more starter or rumen and have more rumen development prior to weaning. So I'm unclear if there's a large response with respect to lifetime performance through early rumen development. But a very interesting question. Dr. Mark Hill, you mentioned a smooth transition from milk to dry feed in weaning process. Many wean from 2 to 3 pounds of solids in 7 days. This is not smooth. How would you consider a smooth program? Other than the Sweeney paper that was doing an automated feeder, and this was out of UBC, there haven't been a lot of studies looking at this transition during this weaning period on accelerated feeding programs. And the recommendations from that paper were 10 days. And I think that paper's is a little bit different because they were weaned at around 40 days of life, and I think it will definitely change with respect to when you're weaning them based on the paper that I published in Journal of Dairy Science last year looking at age of weaning. But I think the later you wean, the better, and you can get away with a 7-day step down. But if you're weaning really early, I think you're going to have challenges even with a 10 or a 20-day step down because these calves aren't going to be getting enough calories during the step down period. And I would say the longer that you can have a step down, the better. Even if I've known some of the best herds I've worked with are doing step downs of up to 4 weeks, and it works amazingly well, but they are weaning later. And I think a big component of this is the age of which the animals are weaned and the success of your step down program. I think it becomes less critical the older the animal is. And I would definitely recommend over 2 weeks if I was recommending on a farm. So that's what I would consider a more smooth transition. With automated feeding, it makes it a lot more smooth. And at our research farm, we're doing a 2-week step down, and we're gradually, every day, reducing the amount of solids. So Carla, first, thank you for your presentation. You previously said that maybe we are limiting feeding during post-weaning. And if an increase in the amount of dry matter, do you think that we will have the same feed efficiency? So I'm just basing it on about 2,000 measurements that I conducted on farms, as well as some controlled experiments that I did with Trevor DeVries at the University of Guelph. And the feed efficiency we noted from those experiments and some of the experiments that we were monitoring on farm was around the 3.5 to 1 mark. So 3.5 kilograms of feed to 1 kilogram of gain, which I had to double check because I didn't believe initially, but yeah, that is in fact where the animals are, at least when they're being fed elevated elevated feeding schemes. So I think that we need to study that a little bit more. If I recall even recommendations when I was a nutritionist being, let's feed 2 to 3 kilograms of a grower post-weaning and then free access to a forage. And in my opinion, in my experience in measuring post-weaning forage intake, they're just not consuming as much forage as we think, and we could potentially be limiting them. So I think the feed efficiency is there. It obviously depends on the proportion of concentrate, but if you're feeding a heavy concentrate diet post-weaning, the efficiency will be quite high. So I think that's the end of all the questions that I've received. Thank you for your positive comments, and if there's any questions or if you know of any graduate students that are keen to learn more about calf physiology, gut development, microbiology, and molecular biology, or even transition cow work, please send them my way. I'm aggressively looking for high-quality students that are very passionate about what they do. I'm sure there's many of you on the other line, on the other end of the line. So please feel free to email me if you have any questions. Yes.
Dr. Steele, there was one last question here, and it was at the very top, but all the other ones were pouring in, so it got buried. But Emily was asking, what kinds of antimicrobial treatments are you interested in studying in calves?
So a lot of my proposals are including oxytetracycline and the effect on the gut microbiota and also metabolites within the digesta, and ultimately gut proliferation and barrier function. So I am interested in oxytetracycline because it's a broad-based as well. It's the most heavily used antibiotic in pre-weaned animals, at least based on the milk replacer companies that I work with nationally and internationally. That's the one that they're using most of. And as for alternatives, I'm very interested in, I've done work with particular new strains of probiotics and also looking at different plant extracts as well and the effect as an alternative to antimicrobials. What's kind of different about my approach would be I am very interested in the short and long-term impact of the microbiology of the gut, but also what happens to the actual gut. Because you are going to be altering the microbes, but you're also going to be altering the output of metabolites in the lumen through microbial digestion.
Well, terrific. Thank you very much, Dr. Steele. Seeing as there's no other questions, I want to thank you once again for your time today. It was an excellent presentation once again. We're most grateful for your time taking it out today and sharing some of your research with us. As for the other attendees, I'd like to extend my appreciation for sitting in on this and asking some great questions. If you have any others, please send that to Dr. Steele in his email, as well as let's try to give him some recommendations for some awesome graduate students.
Great, and thank you very much. And I really appreciate this opportunity. And if you have any more specific questions, I didn't present a lot of my molecular and microbiology results, but I'd be very happy to share them with people if they're very interested in them.
Terrific. Thank you very much.